Today, we're going undercover. We're diving into a world that's totally invisible to the naked eye, but it's filled with some of the most ingenious and dangerous criminals on the planet, bacteria. We're about to open the case files on their secret weapons and their signature moves.
It's a really good question, right? How does something so tiny become a master criminal? Well, we're not talking about simple germs here. These are highly evolved organisms with incredibly sophisticated strategies for survival and attack. Each one has a unique modus operandi that can outsmart our body's defenses and even our best medicines.
So we're going to take a case file approach to investigate 5 of the most notorious bacteria out there. We're going to break down the precise, almost clever mechanisms they use to cause disease. Get ready for some serious microscopic detective work.
First up, we're looking at the internal assassins. These bacteria don't just launch an attack from the outside. No, they infiltrate our very cells and unleash toxins that destroy them from the inside out. It's a strategy of pure cellular sabotage.
Our first culprit is Corynebacterium diphtheria. This is the agent behind the deadly disease diphtheria, and it's an absolute master of biochemical warfare. It relies on a single, devastatingly effective toxin to do all its dirty work.
So what's its secret weapon? It's a process called ADP ribosylation. Now, think of it like this. Imagine you're trying to stop a complex machine. You could blow it up, or you could just stick a tiny piece of gum into one critical gear. The machine is still there, but it's completely disabled. That's exactly what this bacterium does to our cells.
The precision of this attack is just remarkable. The diphtheria toxin gets inside a cell and it hunts down one specific vital protein called elongation factor 2, or EF2. Now, EF2 is essential for building all other proteins. Using that ADP ribosylation trick, the toxin just shuts EF2 down. The cell's entire factory grinds to a halt, and the cell dies. It's this widespread cell death that causes the awful symptoms of diphtheria.
All right, next on our list of saboteurs is Pseudomonas aeruginosa. This is a really versatile and opportunistic pathogen. You might find it causing trouble in chronic wounds or leading to severe pneumonia. And its method? Well, it's less about quiet sabotage and more about pure, brute force destruction.
Pseudomonas produces this incredibly powerful toxin called elastase. It's a type of enzyme called a metal protease, and it acts like a molecular acid. It just chews right through structural proteins like elastin and collagen. You know, the very stuff that holds our tissues together. It doesn't just poison a cell, it literally dissolves the structure around it. So let's just put this side by side. Both are saboteurs, but their methods couldn't be more different. C. diphtheria is like a silent assassin, using a very specific biochemical trick to stop protein synthesis from the inside. P. aeruginosa, on the other hand, is a demolition expert, using a powerful enzyme to physically tear down our tissues. 2 culprits, 2 totally distinct but equally deadly strategies.
But you know, some bacteria don't just focus on the attack. Their real genius lies in defense and evasion. These are the untouchables, bacteria that have developed incredible ways to hide from our immune system and, even worse, resist our most powerful drugs.
Okay, case file number 3. This one is a major threat in modern medicine, Klebsiella pneumonia. Specifically, we're talking about carbapenem-resistant strains. These are a huge challenge in hospitals all over the world.
The main reason this bacterium is so terrifyingly dangerous is its ability to produce enzymes called carbapenemases. Think of these as molecular scissors. Carbapenems are a class of incredibly powerful last-resort antibiotics. They're the big guns we pull out when absolutely nothing else works. And this bacterium has evolved a weapon to simply find them and cut them apart.
And this bug has a whole defensive arsenal. Its primary weapon, number 1 with the bullet, is making those carbapenemase enzymes, the scissors that destroy the antibiotic. But it doesn't stop there. It also has secondary defenses. It can ramp up these things called efflux pumps to physically kick any antibiotic out that manages to get inside. Or it can just reduce the number of pores in its outer membrane, essentially locking the doors to keep the drugs from ever getting in. It's a multi-layered defense that makes it incredibly tough to beat. Let's turn to our next untouchable, a true master of stealth, Coxiella burnetii. This is the culprit behind a condition called Q fever, which can sometimes lead to a really serious heart infection.
And here is its signature crime. It's the leading cause of something called culture-negative endocarditis.
What that means is a patient has a severe infection of their heart valves, but when doctors take a blood sample and try to grow the bacteria in a standard lab dish, nothing shows up. The culprit is there, causing damage, but it's invisible to standard detection.
So, you can imagine the problem for doctors. How do you fight an enemy you can't even find? It's like a ghost in the machine. It's causing all this damage, but it leaves no traditional fingerprints at the crime scene.
So how does it pull this off? The secret is that it's an intracellular organism. It doesn't just float around in the bloodstream, it invades and hides inside our own cells. This is its ultimate disguise. And because it won't grow on a standard lab plate, doctors have to turn to more advanced techniques, like looking for antibodies in the blood with serology, or detecting its DNA with PCR, just to prove it's even there.
Okay, our final case file introduces a different kind of criminal. This one isn't famous for its stealth or for a specific toxin. It's known for the unique, unmistakable evidence it leaves behind at the scene of the crime. A calling card, if you will.
Meet our final culprit, Actinomyces israeli. This bacterium causes a chronic, slow-moving infection called actinomycosis, which can form these deep abscesses pretty much anywhere in the body.
The unique calling card left by actinomyces are these things called sulfur granules. When doctors drain an abscess caused by this infection, they find these tiny, hard, yellowish specks. For a long time, spotting these granules was the key clue that told them exactly which bacterium they were dealing with.
But here's the most fascinating part. Get this, the sulfur granule is a total misnomer. It contains no sulfur at all. What it actually is a hardened macroscopic colony. It's a tangled mass of the bacteria themselves, all clumped together with cellular debris. So the calling card isn't something the bacteria makes. The calling card is the bacteria, organized into a visible clue. How cool is that?
So let's quickly recap our 5 case files. We've seen sabotage by jamming a cell's machinery. We've seen brute force tissue destruction. We've seen incredible defenses that neutralize our best antibiotics. We've seen next-level stealth by hiding inside our own cells. And finally, a unique signature left right at the crime scene. Each one demonstrates a remarkable and totally different strategy for infection.
And that brings us to a final, really crucial thought. This is an ongoing arms race. For every new drug we create, for every new diagnostic tool we invent, bacteria are constantly evolving new ways to sabotage, destroy, and evade. It just goes to show you that evolution isn't always about brute strength. Sometimes it's about information and ingenuity. It really makes you wonder, what terrifyingly clever new strategies will we be investigating in the case files of the future?